protein stuff. Yeah.
Use it for imaging, or what do you use it for?
Yeah. I mean, well, there's a lot of things. There's a lot of atomic physics and things like this, and but actually on the biology side, it's interesting because you can combine it with terahertz, very strong terahertz that's also generated from the FEL itself, and that's perfectly synchronized with the X-rays, and you can sort of do. I mean, people are looking at smaller proteins and or amino acids and things like this, and sort of looking at their responses. I mean, things are more in the gas phase because because of those softer wavelengths. Yeah, but it's I would say it's a lot of more towards the atomic and sort of optical sciences.
I see.
What are the next FELs coming online?
Yeah, the next ones. So Shanghai is building a big one. Not sure when that will come online, but they're going really, going fast at it. Korea has opened up in the last couple of years, so that's one of the newest on the block, and the Swiss FEL as well.
Yeah.
So they're really good, and they have a special beamline just for serial crystallography, and so they're really into it.
Very good.
Well, now we're almost there. Oh, no, there are still 2 minutes to go. Okay,
Yeah.
so we. So how many facilities are there now worldwide? It's sort of 4 or 5 already?
Yeah, something like that. There's the one in Stanford, which was the first hard X-ray facility, LCLS. Then the next one to come online was Japan, Sakura, near Spring 8. And then I guess who came on next? It was either Swiss FEL or the Koreans. Oh no, the European X-FEL must have been the one in Hamburg. Yeah, so that's 5 that are that do real hard X-rays. And then there's 2 soft X-ray FELs, so also the Flash in Hamburg and Fermi in Trieste.
Trieste, right? Okay, that's 6 already.
Yeah.
So I think we give it another minute, huh, Gilad? And then
Yeah.
Yeah,
Forget to send me this slide at the end of the talk.
Yeah. By the way, I shared my screen. Do you see the slide?
Yes.
Yes.
Okay.
So I'm looking for all the friends in the audience. I see that Dave Tirumala is there.
Yeah, I've seen Susan.
Alicia, Andreas Pluktum. Many more. Phil Unfinrod, we just talked about it.
Ah, good. I was hoping you would join.
Yeah. Okay, looks like the number are still increasing, numbers of participants.
Hi, Gilad and Henry. Yeah,
Ah, hey, Charles.
yeah.
Nice
Yeah.
to see you there.
Hi. Good to
Good.
see you.
Hi, everybody.
I can.
Hello, hello.
I can.
Yeah, well, Hagen, maybe we can slowly get going with the introduction. But you need to unmute yourself.
You're mute again.
I'm reading tons of people and nobody hears me here. Yes, I'm mute. Okay. Thanks for telling me. All right, so shall we start?
Yes.
Yeah. So welcome everybody. It's nice to again see so many of you for today's webinar on protein folding and dynamics. It's actually the 13th talk that we have in this series. And our program, at least on the website, now goes until end of January, but I'm pretty certain there are more talks to come, even beyond the schedule that is published on the website. So I'm really looking forward to that. I think when we look back the past 10 years, there were certainly two methods that really widened possibilities in structural biology. One certainly was cryo-electron microscopy and the use of direct detection. The other one, I think, was serial crystallography using coherent and very short X-ray pulses. And it really gives me a great pleasure to welcome Henry Chapman today in our seminar. Thanks a lot, Henry, for being able to make it. Henry Chapman is one of the leaders in this field. He was among the first who has used a free electron laser at the time at Stanford to determine the structure of a large membrane protein complex, photosystem 1, just using very tiny, well-hydrated nanocrystals, crystals that were way too small for standard crystallography. And by now, he's actually leading the X-ray imaging division at DESY, at the German electron synchrotron, where he's developing algorithms, instrumentation methods for structure and structure determination, and also imaging using free electron lasers. So Henry Chapman is certainly the leader, or one of the big leaders in the field of using free electron lasers to address fundamental biological questions. And of course, he received many awards and prizes, and I really want to keep the list short, and I will only mention this year he received the Aminov Prize from the Royal Swedish Academy of Science, and he also was elected member of the Royal Society. And now today, Henry will talk about his latest results on serial crystallography and diffraction using X-ray free electron lasers. But before I hand over the stage to Henry, I just want to mention briefly and to announce our next speaker in this series is going to be Yvette Bahar on November 2nd. That's right after the time change. So it's winter time by then, November 2nd, and she's going to talk about network models in biology. And so with this, I would just like to ask each of you to mute yourself. As usual, we will have a question and answer session after the talk, and simply use the chat window, type in "I have a question," and I will call you by name, and you can ask your question to Henry in person. Okay, and with this, I don't want to steal too much time from Henry. Henry, stage is yours. I will stop sharing my screen. We are really very much looking forward to your talk. And thanks for being with us today.
Okay, thanks. Yeah, it's more of a couch than a stage that I'm calling from, but yeah, thanks so much for having me on the show. In a sense, I mean, it's really nice to have this forum in these these times. Also, Hagen mentioned the Aminov Prize, which was really nice, and I want to point out I'm sharing that with Janusz Hajdu and John Spence, who are really part of this development and the story of getting things going. So it's a real honor to share that together with them. Yeah, so I'll be talking more about the methods and how to sort of measure say, molecules of proteins undergoing reactions or following some sort of pathway initialized by some trigger or other, using X-ray free electron lasers. And I'll try and introduce, the idea of the FELs and how they work a little bit. I mean, I know this is a very diverse audience, and so I'll try and give you access to how this all works. But I thought I'd start out just with this really nice picture, which was this is protein RNA complex, the ribosome, which is from Ada Jonat's lab, Ada Jonat's work, and this rendering, which is really beautiful, done by Holmes, who was part of her group and still in Hamburg. He's actually in the building just a floor below me. And so just from the various structures, trapped in various states with particular messenger RNA sequences or whatever, you could sort of figure out how the ribosome works, and just by looking at these sort of trapped states. So that's great to figure out how this molecular machine works, but really there's still a lot of questions. How forces transduced and signals transferred throughout such structures, and how do they really respond to the environment and to other proteins. And for that, it really required going towards two time-resolved measurements in real time, and away from cryogenically sort of captured structures like this. And so Phil is on the line, Phil Effenrud. So he was really the person who led the team that did the real sort of observations of towards chemistry to see how these proteins can respond at these extremely fast timescales that correspond then to atomic, so atomic length scales go with very fast timescales that are on, well, today with the FELs we talk about femtoseconds. With the tools that Phil had at the time, he had access to synchrotron X-ray pulses, which were about 100 picoseconds in duration. And what you see here from their paper is the myoglobin. And here's looking at along the plane of the heme. This was bound not to oxygen, but to carbon monoxide. And this is a particular mutant that responds quite fast and quite readily, and the CO can be displaced or photolysed using an optical pulse, a laser, optical laser pulse in the orange, I think. And what's shown here is the overlay of two structures. One of the ground state before hitting it with an optical pulse. So this is from a large crystal that's placed in an X-ray beam and measured through the method of crystallography, which I'll explain more in a second. So that ground state is shown here in magenta, and also the stick model there. And then overlaid on top of this is a green structure, which is measurements in a synchronized way with an optical an optical pulse that triggers the reaction, and then the X-ray pulse following that precisely at 100 picoseconds later, and then allowing the system to fall back to the ground state and repeating that over and over and building up the signal through Xray diffraction. So where green and magenta sort of overlap in the computer, you get white, and so that's the sort of common structures, and so you can sort of see molecular motions here where you go from magenta to green. So here's the CO leaving, and then there's a response, or at the CO sort of moves over to here, and this group moves across, and there's a lot of coordinated motion, and a water disappearing over here, And here's a movie showing over various time steps, showing that that motion, and again where the ligand sort of ends up through actually different complements and things like this. But the interesting thing here, which Phil really pointed out, was these are not states that you can really freeze trap and then measure sort of afterwards at your leisure. These are the really strained, far from equilibrium kind of systems, and the only way to really capture them is in time, by watching that process and taking the measurement exactly at the right time. And the ability to do this then depends on your ability to do that synchronization, and also the exposure time that you have available in the X-rays. And of course, you need a certain number of X-rays to give you a signal. If they're in a very short time, this means more X-ray power or brightness, and it really requires these new modern sources. Just so then, the logical extension from that pioneering work by Phil and others, once FELs came online, was to really carry that out at femtosecond timescales, which is really the timescale of the isomerizations and the first moments of photolysis. In this case, this was work led by Marius Schmidt from Wisconsin, and it's been really fun to work together with Marius on various of these measurement campaigns. He's been looking at this photoactive yellow protein, or PYP, which is a real model system for such time-resolved systems. And there's a chromophore in here where you see all the action happening. And instead of Phil's sort of green and magenta, we've got blue and red, but this is actual difference density, so it's showing positive and negative densities. But that's essentially sort of the same. But so again, you see that there's a lot of motion happening in here, and you can taking again the same sorts of snapshots in a pump probe experiment. You can follow the structures. I mean, you can actually refine individual structures. And here, looking at the trans to cis isomerization of the chromophore. So actually a sort of rotation or torsion around this C2 atom here, and tracking that angle. You see there's a very fast response. And let's see, I think, yeah, I've got a movie here just from the sort of showing how that takes place. There's actually a sort of two kind of rotations that occur there, and then you can even at longer timescales sort of follow the response of the protein. But it's very sort of rapid and coordinated. So really, that then you have access to these initial responses, photochemical responses of proteins, and can that are these conformational changes in the system that's driven by light. But you can also then follow at much longer timescales to see these coordinated motions, and even try and track functional changes that are driven by these systems. And of course, photoactive systems are great for this because you have this very precise way of triggering them. But of course, we'd like to do this more generally, or find ways to follow other reactions. But I guess the photochemical reactions are sort of the ones that are easiest to measure and give many insights into protein dynamics, of course. So in terms of how to measure these kinds of changes, as I said, we want to be able to do this on sort of not cryo-frozen systems, because obviously they're not at the physiological temperatures and won't be going through these processes, or if you capture an intermediate, you've obviously done something else to it to say freeze trap it. So I'd like to do this sort of on the fly in real time, so X-rays are a sort of ideal way to do this, and through X-ray crystallography, or also solution scattering, so wide angle or small angle X-ray scattering, which gives you insights on larger timescales, which naturally means sort of longer, larger length scales, which naturally means longer timescales. And then, as I mentioned, laser excitation is sort of the way to get towards this ultra fast, or to really see things at the ultra fast timescales, and sort of bring you along to these more general reactions. But we can also, at longer timescales, perform mixing experiments, and again, the sort of condition FELs allowed these types of experiments, and I'll just give this example before I sort of go back and look at how we do these experiments. This example of the riboswitch, adenine riboswitch. This is under the leadership of Yunxing Wang from NIH. And here, this riboswitch, it's a non-coding part of messenger RNA that's connected to that which codes for proteins involved in purine and adenine metabolism. And this undergoes quite a large conformational change on the presence of this particular ligands that is involved in this process. And on binding, that changes whether the RNA can be translated. So here, just sort of morphing between measurements at different timescales. You can see, as I said, there's this huge conformational change of this whole sort of foot region, and it's so large, in fact, that if you have a crystal of this aptamer and mix in this adenine, the crystals just tear themselves apart and crack and break and fall apart because of such, there's no room for this to happen in the crystal form. But if you get down to sort of micron-sized crystals, they seem to be much more sort of elastic. They deform to allow these conformational changes to occur, and actually go through a phase transition into a sort of a new crystal form in the new state. And so this seemed to be only possible with these extremely small crystals. And furthermore, we could kind of mix on the fly. Say you have a reservoir of the riboswitch crystals flowing down into a liquid jet system that I'll show a bit more about later, which flows these crystals across the X-ray beam, which is coming through here and giving flashes of light to give rise to the diffraction pattern. And then you can also flow these adenine molecules and let them mix together, and then the sort of the delay time is just set by the length of this tube here, which could be, in this case, very long, and it could be many seconds, or you can bring it down to milliseconds. And so that works also extremely nicely, and has opened up quite a lot of sort of new ways to look at of binding in these and capturing very transient structures. And we sort of envision that such a way could be used for screening proteins against, for example, compounds that for drug discovery and things like this in a very rapid way. Just if you do a sort of all through fluidics and a few seconds of each particular compounds against a sort of a set of against your target, you could sort of just sort of screen many things in a reasonably short amount of time. Okay, so as I said, all these things have relied upon having the increase in brightness of sources, and so here's a plot of the development of X-ray sources since the very beginning of time or X-ray time. So with Röntgen's discovery of X-rays. And over time, you can see that things have really amazingly increased by many orders of magnitude. What I'm showing here is peak brightness in these units with, essentially, it's what you think of as brightness. The brighter it is, the more photons, of course, you have. But this is also essentially how laser-like the light is, so how collimated you can make it, how well you can focus it on your sample, and in what amount of time you get all those photons. And you see that we were sort of down at Holmes sources, not giving too many photons in a short time in a certain direction, until accelerators came along. So these were synchrotrons, such as shown here. So it's actually a storage ring that stores electrons and keeps them going round and around. And of course, you may know that if you have an electric charge and you accelerate it, it emits radiation. And making things go around in circles is an acceleration, so it sprays out X-rays in all directions. Initially, these were sort of parasitic to the particle physicists who built these machines to start with. And that's why a lot of the sources are in sort of what were traditionally particle physics labs like Stanford and Daisy, where I am. And but soon, the particle physicists got tired of these guys camping out and using their time and X-rays, and people built on-purpose machines for generating X-rays, and then they got even better with third generation sources that made special devices to really amplify the number of X-rays coming out with undulated devices. And this progression on this log scale is much faster than Moore's law. I mean, it is exponential. And then, XFELs came along, and there was even a huge bump of another billion times in peak brightness due to the fact that you could sort of squeeze all these photons into a very short time. So just to sort of give you some of this history here. So this is a nice picture here of Ken Holmes, who was one of the first people to make use of these parasitic synchrotron radiation, and he used it to look at frog muscle fibers and do fiber diffraction, and it was amazing. You got 10 or 20 times better than a home source in those days, and you could sort of stand here with the X-rays. And but this also then Keith Hodgson over at Stanford showed you could do protein crystallography much better than you could do with an X-ray tube. But even so, 5 hours exposure time to get a decent measurement of this ribberexidon crystal in 1975. So of course, time resolution wasn't really on anyone's horizon in those days, but it's amazing how things developed. And just to give a flavor of the development of crystallography, here's a plot of the number of depositions in the protein data bank, the cumulative number growing over time. And so number of structures, and we're here in 2020. And blue are the X-ray structures, and that's really took off in the mid-1990s. And again, that was when sources like the National Synchrotron Light Source in Brookhaven came online with undulators. You know, there was also dedicated beamlines, detectors, software, all that really automation made things really exponentially increase. Well, more recently, it's more of a linear increase. On top of that, you've got NMR in this gold color, and we've heard in this seminar series, for example, about NMR spectroscopy, and also as Heiger mentioned at the start, cryo-EM, which was sort of a little sliver. But if we go here to 2020, you see there's actually a couple of thousand structures in the PDB from cryo-electron microscopy. And if you compare that with the number of sort of the jump in X-rays from year to year, which I guess would be about here to here, you know, it's catching up. And I would say, you know, cryo-EM is essentially where molecular crystallography was in 1995 or so. And you can see, you know, it's ready to really take off. Perhaps, of course, you know, it's a bit of a different game with electron microscopes in labs rather than these big facilities where people come. I mean, yeah. So there's it'll be interesting to see how this develops. But of course, yeah, I just wanted to point out it's not just the numbers, you know, it's all these beautiful structures. And up until kind of the FELs came, most of these structures were static, you know, except for these pioneering experiments of Phil and people like Keith Moffat and Janusz Hajdu and others. Okay, but I wanted to, because yeah, set the stage, so to say, as Hagen was mentioning earlier before we started, and talk a little bit about X-ray diffraction, what we're doing, and to also give a bit of an appreciation of where this can go, right, with these very intense laser-like beams that we can get from FELs. And actually, one of my students printed this very nice sort of pattern, and you can shine a laser pointer on it, and you get these brilliant diffraction patterns and things. And it's great to sort of demonstrate this live, but here on Zoom, it's better to go with the computer version. So here are two slits, say, in a screen, and if I had my green laser pointer and looked on a screen in the distance from the light coming through these two slits, we'd get this nice interference pattern. So essentially, the two expanding waves overlap, and in some directions, you get strong constructive interference, and you get bright lines. In other places, the waves actually come out of phase with each other. So whenever one's at a peak, the other one happens to be at a trough, and you get light just cancels and disappears and gives you black. So it's this is what we're relying upon. Actually, all of imaging essentially relies upon this, whether you have a lens or not. But here we don't need a lens, and yet we can sort of somehow infer structure from this diffraction pattern. And in fact, if these slits were further apart, these two waves essentially come at each other at a much steeper angles, and the distance between these light and bright, these fringes becomes smaller. So there's this reciprocal nature between the distance of these slits and the frequency or of the fringes. Now, if we made the slits actually go together really closely, you know, then essentially they would merge into one, and the closeness, you know, that's essentially given by the wavelength, or how close things have to be before they're indistinguishable is the wavelength. Or conversely, to see, say, two atoms which are only an angstrom apart as sort of the source of two waves, you need X-rays, of course, to sort of have that scale. So here's a calculated diffraction pattern And here we see lots of these fringes, and there's fringes of different period, and you can see it's sort of finer if you go up and down than left right, but there's interesting variations and structures all over the place. And we saw that, you know, fine fringes mean things that are far apart, and broad fringes are things that are close together. So we can actually analyze all this in terms of that by doing a sort of frequency analysis, right? So we're looking for, you know, the fine frequencies or the broad frequencies. So to do a frequency analysis or a spectrum of this spectral analysis, we do that just by the Fourier transform. That's a way of analyzing the spectral content of this diffraction pattern, right? So we have, so we can just do that. And here's our spectrum of our measured diffraction pattern. So we've got, you know, instead of an audio spectrum giving just in one dimension, we've got 2 dimensions here. So we've got our left-right frequencies and our up-down frequencies. And we can see here that, as I said, there were finer frequencies, so higher frequencies, I should say, in the up-down direction. That these are sort of listed up here. And in the left-right, you only got to a certain highest frequency. Somehow there's this point over here, which is, I guess, a little bit fringes that are a bit angled. And this is the highest, in terms of magnitude, the highest frequency that exists in this whole diffraction pattern. There's nothing higher, which means you know, there were 2 particular scatterers that were as far apart as you know, they were the furthest apart in this particular sample. And so what we have here really is a map of all the different distances between scatterers or atoms, say, in the sample. It's a map of their distances and their directions. So that's all these lines. And of course, this is also known as the Patterson function in crystallography. And what's interesting in this, so this sort of describes all the connections between things. And so the question is, what are those things? How are they arranged? So we know all their sort of pair correlations essentially, but how do you get at the actual arrangement? And that's you know, in diffraction space, it requires you to know the phases. In real space, it just requires you to know the positions of these connections. That's equivalent. So that's just another way of stating what the phase problem is or was. And you can see one way of solving this phase problem is bringing all these connections together to make the most compact object. That's essentially, so I just moved all those connections from this map over to here without changing their lengths or their directions, and just arrange them in a way that they fit in the smallest possible sort of area here. And that is the most likely solution to that structure. And then you can sort of say, okay, here's my atoms, and I can model that and as, say, a double helix. Of course, this was a sort of a toy problem, but you sort of see the similarity to this famous photograph 52 from Rosalind Franklin. Which Crick and Watson sort of got hold of and interpreted since they sort of knew their Fourier transform and what this kind of meant in terms of structure. Right. So I showed this and just to show you what you can now do with free electron lasers, because they have such a sort of pack a punch with so many X-rays. It's a very coherent beam, so you have full coherence across all your entire sample, which allows this interference to occur from any 2 points in your sample. And we can measure these diffraction patterns, continuous diffraction patterns, and solve them essentially through finding these phases, or these in real space, these positions to put all these connections together, or these correlations together. So here's just something that was aerosolized, thrown across the beam of the FEL, some soot to measure all the different morphologies that soot can take on, and this is what it looks like. Then when you phase this diffraction pattern, And here a particular virus particle, the mimivirus, that was again aerosolized and shot across the beam. These are all at relatively low resolution, which is given by the scattering angle, or the highest that this diffraction goes out to. Yeah, so just to follow then, a single molecule, if put into an FEL beam, or if you could measure the diffraction from it, it gives you this nice continuous spectrum of frequencies, which is really describing that structure. But it's, of course, going to be extremely weak because the molecule is such a small thing and you don't get that much scattering from atoms. So usually in crystallography, or the goal of crystallography, the reason crystals are used in the first place is really just to bring addition into play to give much stronger signals. And so you want to do that addition in a very cooperative way. Of course, you want all these molecules to be aligned, and crystals provide that, and there can be many, 10 to the 10 molecules in a crystal, all cooperatively diffracting, and where that diffraction from the individual objects in the crystal adds up, it again constructively interferes, but now in these sort of discrete Bragg peaks. Those Bragg peaks are essentially just sort of amplifying the individual molecular spectrum or transform in those directions. Everywhere else you get very strong destructive interference because of the way random numbers tend to sum together. They tend to always find their way back to zero. And yeah, so this crystallization actually leads to a sort of lack of, or a loss of information due to the fact that you're just sampling at these discrete points. And if we did that Patterson analysis that I walked you through just before, what happens is you get confused as to whether a pair correlation occurs within one molecule, or if it's in one of the atoms is in one molecule and the other in its neighbor. Yeah, so that has of course led to much harder problems to do that phasing, but of course there's ways around that, and this can be nicely solved. Now the other sort of thing that why do we need that high signal? Why can't we just put a molecule in the beam and wait longer? And that's, of course, the problem with X-rays, or electrons for that matter, is that they're ionizing. So it's ionizing radiation and destroys the very sample that we're trying to look at. So here's an oxygen atom with its 8 electrons. We want the X-ray to come in and scatter off that atom and give rise to the diffraction. That's elastic scattering. It doesn't do anything to the sample, which is good. But just the way the physics works out, there's 30 times more probability that a photon will come and be absorbed in the atom and cease to exist as a photon. It's given its energy to the electron, which flies out. So it's ionized at quite high energy and velocity. The atom relaxes, and that is in itself also liberates energy, and you've got these 2 electrons flying out. These electrons have enough energy to knock out other electrons, and that process continues, and you've got this whole cascade of events which can lead to several hundred atoms being ionized just from that one X-ray. And there were 30 or so of those X-rays per elastic scattering. So essentially the situation is pretty bad, in that you're just depositing huge amounts of energy into your poor molecule, and it has to, it gets destroyed, and through this ionization and then heating, radiolysis, a lot of course in a crystal, if you're in a with a solvent, there's a lot of water around, and that's been ionized, creating free radicals that then diffuse around and do all sorts of badness. But of course, if you can that all takes time, right? So it takes time for this series of unfortunate events to occur. And if you can sort of do a flash measurement in a shorter time than all this bad stuff to accumulate, you can sort of see the pristine state. This is just showing measurements on a crystal, a virus crystal at room temperature from diamond light source through Elspeth and Robin Owen. So this ionization, as I said, leads to unfortunate events, including the whole destruction of your crystal and just the inability to measure things and changes in the structure that you're trying to measure. So the tolerance that you have, or the tolerable dose, or the amount of X-rays you can put on the sample is extremely limited. It's only a few scattered photons per molecule at most. Actually, it's if that. So you really need many molecules to allow you to get a measurable signal before you've kind of destroyed your object. But yeah, as I said, we've got this other way around it, which is time, and this is a great sort of metaphor for that. This picture by Harold Edgerton. He was the guy who sort of developed flash lamps that were based on capacitor banks and things, and the standard flashes that are in cameras, to really see things on the microsecond timescale. And at that timescale, you can freeze a moving bullet and see that inertia really keeps things in place for some time. Say the top of this card here, and that's what FELs are. They're big flash lamps. Essentially, they produce pulses of light, femtoseconds duration. And they do that through through accelerating now electrons in a straight line. So you've got to sort of unwrap your synchrotron, and you do that so that you can sort of keep compressing these electron bunches into very in time and in space, essentially, and then converting those into X-rays. And here's a sort of quick view of the European XFEL. So this big yellow thing here is the accelerator. We're accelerating electrons up to about the speed of light, or 99.9%. They go through this undulated device, which has lots of magnetic periods. And now we've got X-rays, and X-rays just fly along, fly for about 1 kilometer, where the beam is finally sort of large enough that it won't burn a hole in the optics that you're trying to use to focus it. So they, we're talking about big facilities, but they're used for a lot of different science, and it's pretty fascinating how these X-ray beams are produced, or how this works, because we've accelerated the electrons up to the speed of light. They go then through this undulator, which is a magnetic structure, sort of periodic magnetic structure. So we've got magnetic fields north and south, and then south and north, and so they're going up and down different fields, which sends the electron left and right. So the electron sort of gets this wiggling. Again, it radiates, just like electrons in an antenna will radiate radio waves. Now we're radiating X-rays, but because the electrons are traveling at the speed of light, and obviously the light they produce, the X-rays, is also traveling at the speed of light, right? These two things move together, and they stay together for all hundreds of meters. And as the this field, the X-ray field, which has been generated, grows, it forces the electrons into little into that structure of that radiation. And now the electrons, when they're in these little pancakes, they radiate together in phase, and that produces even stronger radiation. And because you get this, again, coherent enhancement, and the stronger the radiation you get, the more bunching of the electrons, the more structure in the electrons, and the more structure, the more radiation. So it's a positive feedback, and it just grows therefore exponentially through this whole length. And it's just like, you know, feeding back a speaker into a microphone. You get this shriek, and that's really what we're using. It's just an amplification in this case from noise, and it grows over, you know, each time you add another meter or 10 meters of undulator, you double the amount of X-rays you have. So you just keep doubling and doubling, doubling, and pretty soon you've got huge pulses. And this, yeah, it's just an amazing engineering to align these things across 100 meters to sort of micron tolerances. Yeah, so you get as a result, you know, about 1 million times more photons in a single pulse than you would at a synchrotron in 1,000 times shorter. So that's how you get 10 to the 9 increase in peak power, which is, yeah, as I said, just a game changer, and it's a completely different situation. And that idea of having that many photons in such short a time led János Hajdú and his colleagues to sort of make the proposal that you could perhaps measure enough scattered photons from a single molecule, or put enough X-rays onto it. Of course, we can't change the scattering cross sections. So, you know, how much, there's still that more ionization happening than scattering, and so every atom is absorbing and ionizing, and there's just, yeah, and it's just ripping itself apart from those interactions. But again, that takes some time just due to the inertia of the atoms. And so you've got a kind of little window of opportunity to see things in their pristine state. And so that was really the idea that was driving us and going to the FELs. And when we started, the first experiments we did, we used these little crystals, as Hagen mentioned, the Photosystem 1 crystals, and there were several reasons for choosing those. One was because Petra Frommer produced them, so Petra Frommer at Arizona State University, and she was happy to produce, you know, loads and loads of these things and sort of sacrifice them to the beam without really knowing at all if this would work. And we chose them because they were quite a large unit cell, because we thought we'd use crystals due to the fact that they do have this periodicity, and that periodicity is a kind of reporter of how good or how pristine the structure is, right? Because if the molecule or if the crystal has exploded, it will no longer be the same from unit cell to unit cell. It won't be a crystal, it will just be a mush, and you won't get Bragg peaks, which are that signature of crystallinity. And so we wanted something with a very large unit cell, because we thought we'd only, with longer pulses that they could produce at first, we didn't think we would see much beyond 1 or 2 Bragg peaks. But it worked extremely well, and we were able to measure many diffraction patterns, as you see here. These are just single snapshots, so each crystal has been destroyed by the beam, and yeah, and so you have this flash photographs of each one, and we're extremely happy to see that this went out to high resolution, and realized afterwards that the crystal was being destroyed by the later parts of the beam, and it wasn't a crystal at the end of the pulse. But the time that it was a crystal, no matter how short that was, did generate that these Bragg peaks. So we were able to sort of measure this, even though it's sort of a self-regulating process. And this was done by streaming these crystals in liquid, so they're in at room temperature, very tiny crystals in these first experiments below a micron. This whole liquid jet technology was developed at Arizona State University by John Spence and his colleagues, Uwe Weierstuhl and Bruce Toeck, and that was really crucial to have this working. It was funny, in these first experiments, we had no way of really seeing what was going on. It was just in this vacuum chamber. We had no cameras, and we just sort of aligned things until we started seeing diffraction, and the whole thing worked for a week or so without breaking down. Yeah, and it was beautiful. Of course, you need a detector that can keep up with all these flashes, and the software to deal with these hundreds of thousands of diffraction patterns. That's done through Crystallography is one software suite that Tom White has developed. He's in Hamburg in my group, but runs this and has made this nice graphical user interface recently, so it's even easier to do. And he has these great tutorials on how to do that. And the method has been used quite a lot to measure not only structures from crystals that were sort of too small or too difficult to measure at the synchrotron, but also to do time resolved measurements. Now, one thing that's sort of interesting, what is to look at the kind of landscape of the measurements in terms of the size, say, of the crystal or object here, from very, very small up to or single molecule up to very large crystals. So in terms of, say, volume of the crystal or number of atoms, this is 15 orders of magnitude here, and here on this scale is our exposure. So in terms of number of photons per square micron, say 10 to the 12 per square micron here, which gives you a certain dose. So you've got dose on an equivalent scale, or number of scattered photons per atom. We'd like to get up to this, sort of teragray dose, which is sort of, as it sounds, pretty terrifying. But yeah, but there you would get as many photons scattered as you have atoms in your sample, which we think is enough to, or more than enough actually, to do measurements from single molecules. Now the product of sort of the number of scattered photons per atom and the number of atoms, of course, is the number of scattered photons. So this is the strength. So this is a sort of constant strength of diffraction. So whether you have a very strong crystal and a weak source, or a very weak crystal or molecule and a strong source, you sort of get the same number of photons, right? And you need about this many to get a 10 to the 10 to get a good structure. And so where serial crystallography allowed us to go was really down, of course, in object size, which was the goal, but we also end by increasing the dose, but we could do that by also decreasing the signal that we got per sample, because we could do this over and over again at quite a high repetition rate. So now many thousands of measurements per second. So that's sort of going to where you'd want to with single molecules. Yeah, so it's getting close, but you can see that there's not many scattered photons per molecule. But at the same time, we saw that we could also sort of not necessarily reduce the size of the sample, but reduce the dose, so that you could avoid the need to cryogenically cool the sample to which extends the dose tolerance, but then reduces the temperature, obviously, and also sort of push things towards to do time resolved measurements at synchrotrons a bit more readily, and now sort of optimization is a matter of how much signal you get versus how much noise. Obviously, it gets more difficult as you're down this end, because a very weak scatterer compared to some background scattering is hard to sort out. So you need to be careful. It gets more and more sort of careful how you introduce the sample into the beam. It's also interesting where cryo-EM sits in terms of scattered, say, electrons per atom. So it's about 1/20th of the atoms in a molecule will give rise to the signal in the image, and so that's sort of around here. So we should again be able to use the similar techniques, although in diffraction space, and recover structures hopefully at room temperature. As I said, there's different ways to do this, but I think I'll move on and show you more the liquid jets. So these liquid jets are just shooting these microcrystals across. Here's a little glow from the X-ray beam, creating a plasma. And this is on these PYP experiments. And simply by just shining the laser at this region and timing that just in the same way that in Phil Affenrud's experiments, do this pump and probe measurements, you could sort of then follow these series of measurements or at this series of times. And here, because the crystals are so small, you can sort of efficiently illuminate their entire volume and get very good sort of yields. And so therefore that was easy to sort of bring it to the femtoseconds. And just to give some of the statistics here, so quite good resolution, and with many hundreds of thousands of patterns. And now it's possible at the European XFEL, which has this high repetition rate, to measure, a few, well, certainly tens of thousands of patterns in tens of minutes. So this works quite well. I think I'll, and I mentioned before the mixing experiments. Here again, you'd sort of want to do this mixing quite rapidly. I just and Xray Knoska in my group, a graduate student in the group, also working with others such as Michael Heimann, who's now in Stuttgart, developed sort of these microfluidic mixing devices to sort of bring crystals and ligands close together, and then to the point where you just want to be limited by diffusion times into these very small crystals, which should be sort of microseconds. Yeah, so that you can sort of flow these two liquids through, and he was characterizing this through X-ray tomography. So using one dark liquid, say iodine and water, and just flowing this through and putting it in an X-ray beam and now taking tomographic slices at different depths through here, which corresponds to different times. You can sort of see how quickly these liquids mix, and it's it's obviously you have to take care about mixing ratios and times, and this can be optimized to sort of get fairly good 50% mixing ratio within about 10 milliseconds or so. So that's about the time precision that you can do these types of mixing experiments, and you can build this into the jet. Of course, you can sort of add delay lines and things like this, and it's all kind of like a little mini Lego system. These nozzles were produced by and the mixers produced by 2-photon 3D printing, which is quite fun to work with. But you can also do things that do these mixing experiments, as I said, at synchrotrons quite nicely, where you sort of say mix and write your sample onto a moving tape that just drags your sample across the beam, and you can measure these things or bring them across the beam at the rate that you can measure them, so that you're not wasting sample in between. And that has a very low sort of consumption rate of material, which has been a sort of problem in many of these experiments. You can even sort of pre-write your tape with, say, the ligand and put your sample onto it for faster timescales. So that's been developing quite nicely. And again, because I don't want to run out of time. And going sort of back to Phil Affenrud's measurements, where he used broad bandwidths to give you many more photons. It's really possible to do these types of experiments with single pulses at the synchrotron. So do serial crystallography at 100 picosecond exposure times with reasonably sized crystals. And because the broad bandwidth gives you full sort of view of each Bragg peak, you sort of get this integration through the Bragg peak. You don't need that many snapshots, in fact. And so you can do this in a few in quite a few crystals. You can, and if you have now a detector like the Jungfrau, which was built by Detris for the PSR people for FELs, then you can do kilohertz rate measurements at synchrotrons, and so things are just getting sort of faster and faster, and then you're sort of limited by your computers and how much data you can digest. Right. So coming back to here, and where are we going? How likely is it to meet the dream of measuring single crystals? These were just simulations here. So experiments are going on with measuring single particles, quite large particles to start with, such as viruses. And the key we've realized has been in getting the scattering background low, so that you can sort of trust that these little dots out here, which are single photons, really came from the object, so that they can be correlated and merged with other diffraction patterns in the same way that cryo-EM sort of correlates and merges images. Now, the signal-to-noise ratio is around 1 at a resolution, or at about 5 nanometers. And that's actually similar to what single images of cryo-EM have. Cryo-EM images usually look like nothing much at all, but there is information there, and that's the whole key. And more recently, so another problem has been just getting enough of these diffraction patterns. So we need to use a very focused beam to get the highest intensities, and then most of your particles you shoot through kind of miss things. But with now nice software and the detectors and the beamline, this is now the European XFEL. You can start to optimize and track things. So here's a real-time display on sort of the fraction of pulses that are hitting, giving rise to nice diffraction. So you can sort of tune it up nicely in a short time. And this classification and averaging also works really well. This, of course, well, it's not protein. There were little gold particles, but in an experiment led by Kartik Iyer in Hamburg, and many others, and also Xavier in my group making these particles, we're able to sort of classify diffraction patterns of similar nature, to average them up, and build up, from about 10 million patterns, all these different classes, and reconstruct these. So just showing you the sort of view of the diffraction here, and also the view of the reconstructions of different samples that were put through. I mean, they're very sort of simple and not very interesting from a dynamics point of view, for sure. But these yeah, the reason we chose gold was because for a larger beam size, that would give us many scattered photons as we'd expect from something like photosystem 1 here, in a much more focused beam. And this is shown here to scale. Of course, it's not just size that matters, but the scattering strength of the atoms are also weaker here. So it's a bit more challenging than maybe it looks. But we're sort of fairly confident that if we can, do better with the way we can introduce these samples into the beam, we can basically do those types of experiments and map out the variability of conformations over quite a large space. This again just showing on the gold, but now imagine we're doing this on proteins and sort of capturing different events here, maybe even sort of some rare events as you sort of go through your transition state, for example. So I think there's a lot of scope and promise in these sources, there, and I sort of emphasized that future work because that's what's driving us at the moment. But there's also in the serial crystallography, a real tool that can be used to study protein dynamics, and many ways to go about it. And I didn't speak about all those ways, but many ways to trigger reactions quite precisely with the FEL pulses. The facilities are coming online. People asked how many. There's 5 FELs now in the world, more to be added, such as the Shanghai source. Synchrotrons are also getting brighter and brighter still. All their upgrades that are ongoing, and it's just getting more and more capabilities at those sources. Right. So I just and there by sort of showing a photo of the group and people who've been involved in this work. So it's been a really pleasure to work with them, but also many collaborators around the world. Especially at ASU, SLAC, Wisconsin, and New Zealand, Sweden, France, and Ned in NYU, and the guys at the FEL. Yeah, so thank you.
Well, thank you very much, Henry, for a terrific talk and terrific insights. There were already questions during your talk. So the first question was by Miranda Lynch. Miranda, are you still with us?
Sorry, I had to get unmuted there.
Yeah.
I believe some of this was covered actually right at the very end there, but one of the things I was curious about was just sort of the sample requirements. It certainly seems like, even with sort of the deposition on tape and so on and so forth, you're still looking at, some pretty serious need for sample, as well as whether you could comment on the sort of size distribution that's allowed for these injection systems for the particle distribution. I am very interested in detecting the sort of smaller nanoparticles, and was wondering how you kind of achieve that.
Yeah, I mean, so I've been very sort of aware of the consumption because it's been, especially the first experiments, it was very difficult for people to to meet the needs, and they had to go into sort of full-scale production and lots of grad students working around the clock to get all those samples. So we've been really working to reduce that, and the tape, as you mentioned, is one of the most conservative. I would say it's for a full measurement you only need tens of microliters, maybe of sample, where you've got maybe a volume fraction of your particles around 10%. So, I mean, maybe for some cases where especially if you've got to extract protein from natural sources, then that could be still problematic. But it is getting close to sort of the amounts that you have in crystallography trials and stuff like that. But that's really on the conservative side. Otherwise, also if you can put things on a chip or some sort of substrate and scan that around, you can really try and hit every, somehow pre-characterize that and go to where the crystals are and make best use of what you do have. Whereas the liquid jets, you waste probably, you sort of use up 10 to 100 times more material than you actually end up measuring, even now. And we're trying to sort of, again, sort of bunch this or space the particles in there. But in terms of particle size distribution, actually we're quite we can have quite large distributions in the sort of software processing. you kind of average over all sort of sins in the experiment, and there's a lot of things that can fluctuate, and including the X-ray beam, especially in an FEL, it bounces around sometimes. And yeah, so crystal size, that's fine. You kind of just, again, if you're doing 10,000 measurements, things tend to average out. But it is good to have to make that more uniform, especially if you're squirting things in a jet. It's good to not have too bigger samples or too bigger crystals, because they might get stuck somewhere and clog up the works. So we usually put a filter in or pre-filter. People have, with in vivo grown crystals, which crystallize in cells. Sometimes they can be sort of long and narrow, and people have found it's good to actually kind of crush them, or vortex them, break them up into smaller ones. Many crystallographers are horrified when we say that, but it seems to work.
All right. All right. I think so. The next question is by Shaul Mukamel. Shaul, I see you.
Yeah, we've seen.
Unmute yourself.
Okay. Thank you for the spectacular talk. I have a question about the curve that you showed with the PYP. You had the time evolution, and I saw data before and after, but not during the process. Is the smooth curve an interpolation, or you have real data during?
Right,
yes, here, right.
Yeah, that's an interpolation. In fact, the sort of the jitter on the measurement, once you sort of do this, well, we actually measured that on each pulse, but I think there's a residual jitter that's kind of on the order of 50 femtoseconds or so, which is sort of similar to the time here. So I think that would give problems in sort of capturing sort of that real transition state, but that would be yeah.
Okay. Yeah, thank you. Yeah.
Next question is by Ben.
Thank you very much, Henry, for this beautiful talk. I have a question regarding your elegant microfluidic mixer. I was wondering what's the ultimate limits there in terms of time resolution, because that would probably really be one of the most universal tools to study many different kinds of reactions. And so for regular spectroscopy applications, the fastest mixers are in the tens of microsecond range. But I guess for your suspensions, viscosity is a big problem. Or what's the limiting factors that slow you down into the millisecond regime?
Yeah, so it is in one case kind of the volumes that are involved. Although if you sort of make things faster, you can get things going. I mean, if you flow faster, you can also mix faster, but then you're consuming more and wasting more samples. So that's a little bit of a concern. But it seems like there are methods to go even faster than this 10 milliseconds that we've shown. I mean, in microfluidics, there's like these cell sorters and things. You can sort of use the inertia of particles to sort of let them fly into a, or change direction, or fly into a region of different composition, liquid of different composition. So it may, I think there would be a next generation of mixers that will maybe get to sort of microseconds, which seems to be, I'm sorry, below maybe a millisecond. But then you've got the diffusion time of the ligand into the crystal, and that is very dependent on the crystal itself. The sort of solvent content and pores and sort of the labyrinth ways you can get into the crystal. Yeah. So the ultimate, of course, where we'd like to somehow go is to have your single molecule and bring your ligands together. So if we can do single molecule diffraction in liquid, which by reducing the jet thickness down to about the thickness that you have in cryo-EM, again, you should have similar signal to noise in terms of background scattering or what contributes to background in a cryo-EM image. So I think that's encouraging. And now if you've got these flowing systems, even shooting a second drop onto sort of having this collision in space as the X-ray is coming along, then sort of all the dynamics of that could be still I mean still in the hundreds of microseconds, I would imagine, but.
Thank you.
Maybe one follow-up to that. So has anybody done or tried to use caging compounds and then combine it again with flash photolysis? Because that would seem to be the easiest way of getting quick release.
Right. Yes, people have done those types of experiments, and there's a lot of development of developing cage compounds. Just trying to think of examples, but there's been work at the XFEL, actually, very nice work. Even as simple as sort of changing pH, sort of a caged ion that's does that very quickly.
I see. But the limiting factor is the chemistry, basically, developing the caging compounds.
Yeah. I mean, people like Arwen Pearson in Hamburg, I know, is doing this very actively, and there's a lot of synthetic chemistry involved, and trying to figure out where to put the cage so that it doesn't have to travel very far, and what the the release rates are, and so this. Right. Yeah, but it looks
thanks.
Looks pretty good.
So next question, Gilad.
Yes, I was wondering if it would be even possible to think about an experiment where one could image directly the transition state of a reaction. For example, in rhodopsin, where the laser pulse will put you immediately around the transition state in the excited state, and then you would be able to image it on the femtosecond time scale.
Yeah, I mean, this gets to Scholl's question, I guess, of trying to sort of see what's happening between trans and cis. So of course you need a short enough optical pulse, to be before that happens. I mean, things do seem to I mean, at least in that PYP case, evolve fairly coherently. All molecules doing the same thing in time. So that should allow you to see this in an ensemble measurement like a crystal. So that was if things start to dephase, or if each molecule is slightly off in terms of their timings, then there'll be a broadening due to that. Maybe you won't sort of catch it. I don't know. I mean, how it depends also. What's the time window that you have to get a transition state? Maybe it's one of these quantum mechanically impossible sorts of things. But Scholl would sort of know more about that.
Okay, next question by Philip Banfenrud.
Thank you, Henry. This is really some beautiful stuff that you're talking about. And as you're pushing the frontier of this, you show this graph where you're showing if you can get to really high peak powers, you can push toward this single molecule limit better. So I was curious, what's the state of the art now for seeding? Because seeding of these instruments can potentially get you shorter pulses and get you closer to that limit you're targeting. Can you comment on that, please?
Right. Indeed. So most of the facilities, well, or at least LCLS and the European XFEL have seeding in place that can be sort of optionally turned on. So currently they give you higher spectral brightness. So they tend to, they give you a narrower spectral width, but not necessarily more photons and not necessarily in a shorter time. Well, the time I think is a bit more better controlled. So I'm not sure if it's if it's, you know, the big jump that you may think, right? Because you're sort of gaining in this in making sure all the photons are the same color, which we don't necessarily need in the crystallography experiments, because in the end you sort of don't need more spectral resolution than the number of pixels you have across your detector, for example. Yeah, but these machines, you know, the European XFEL has got up to 10 millijoule pulses in their longer wavelengths at a few keV. So that's also, you know, and if you get that down to 10 femtoseconds or so, I think you are around this level, and depending on how you focus. So I think you know, from the source point of view, we're in sight. I mean, there's still perhaps more nonlinear X-ray interactions to worry about, and there you may need to reduce the pulse duration even more to not worry about sort of saturation of scattering or and things like that.
Okay, thank you. But one of the points you made is that when you go to these very weak scattering, background is really the essence.
Yeah.
So while seeding gives you less total photons, presumably they're more coherent and maybe focusable more tightly. But then, of course, matching the sample to the focus of that beam is then the next challenge, right? But so it seems like less could be more if you have better control of it.
Yeah, I agree. I mean, it's better control, certainly that's good.
Okay, thank you very much.
Yep, thanks.
All right. Any other question? At the moment, I don't see any in the chat window. If you have a question, it's time to ask. Okay, so this doesn't seem to be the case. Henry, thanks a lot for a fantastic talk. Really a pleasure to listen and to have you with us. And thanks everybody for joining. See you again in 2 weeks on Monday, November 2nd. Have a great day or night. Goodbye, everybody.
Thank you, Henry. Thanks, everyone.